Attendees are in listen-only mode.
All right, good afternoon, everyone. My name is Tawny Samiski. I'm an entomology specialist with UMass Extension, and I'm excited to welcome you all here today for another one of our invasive insect webinars. So welcome to those of you who have joined us in the past, and welcome to folks who are here with us for the first time. This series is brought to you by a collaboration between UMass Extension's Landscape, Nursery, and Urban Forestry program, as well as UMass Extension's Fruit program. And of course, these webinars are made freely available to you by grant funding support through the Specialty Crop Block Grant Program at the U.S. Department of Agriculture. So we certainly thank them for their support. I also have here with me today Jeffrey Jouet of UMass Extension, who is assisting us with the technical aspects of GoToWebinar. So Jeffrey, if you could please proceed to the next slide. Excellent. So just a little reminder again here for those of you who are looking for pesticide as well as association credits as a result of viewing today's broadcast. All instructions for receiving pesticide credit, which includes mass categories 26, 27, 29, 35, 36, as well as the applicator's license or core license. In addition to association credits for MCAs, MCLP, ISA, and SAF, these will all be shared at the end of this webinar. So please remain on the webinar until the end to receive these instructions and to take the quiz for pesticide credit. This quiz is only required for those of you who are looking for pesticide credit, although folks just taking it for fun are able to do that too. If you sign off the webinar before taking the quiz, you will not be allowed to retake it and will not be awarded the pesticide credits. So again, to those of you who are looking to receive this credit, please remember that. I also want to remind folks who are joining us with their cell phones. We've had a couple of issues, particularly in the last webinar, where folks were using the back button during the course of taking their quiz. So just be careful with that. Try not to use the back button if you're taking your quiz on a cell phone because you may get kicked out before you're able to finish it. So just be careful there. I also want to remind our audience that you have on your GoToWebinar dashboard here a questions area. So you can go into the questions section and enter in any questions that you have during the course of this webinar, and I will try to answer those that I can while the webinar is taking place, and I will save some of the questions that our presenter today will be able to answer at the end as those come in. So again, don't forget about the question function. All right, so without further ado, I'm going to change our presenter here and introduce today's speaker. And of course, we have with us today, we're so thankful to have with us today Dr. Julie Lockwood, and she is the professor and chair with Ecology, Evolution, and Natural Resources at Rutgers University, and she will be talking to us about spotted lanternfly and environmental DNA, insect monitoring of the future. So please take it away, Julie.
Thank you. Just want to confirm my thing that everyone can hear me. I'm pretty sure that's true. Yes, okay. So I will be talking broadly about environmental DNA and what it is, and then showing you cases where we have applied that in terms of spotted lanternfly and how we're looking to expand that into species like emerald ash borer. But I also will be talking about broader issues related to monitoring pest insects for agricultural and forest systems. So this is where the talk is going. I'll start with a very quick overview of the problem we're facing, especially in the Northeast, in regards to invasive insect pests. talk about and explain what environmental DNA is and how it works, again, in the context of insect pests. So that'll be a little bit of time to cross that over and explain it. And then I'll move into the applications of eDNA for insect pests. And I'll show you some preliminary data we have on using eDNA surveys to detect spotted lanternflies in vineyards in New Jersey. And then a little bit on how we're thinking about expanding the use of eDNA into species such as emerald ash borer, maybe other wood-boring insects. And then finally, a really common question sort of related to these techniques is, what is the cost? What does it look like? How, if you wanted to do this, what are the logistical concerns and what are the cost concerns? So I'll address those as best I can at the very end. So we'll start just by, pointing out that we live in a hotspot. We know we live in that hotspot in terms of the coronavirus, but we also live in a hotspot in terms of at least forced insect pests, which this particular map, if you haven't seen it before, includes insect pests of forests and fungal disease pests of forests. And it's just tallied up by county within the US. And you can see that the sort of the more red and into purple you are, the more of those species we have in that county. And the big sort of take-home message here is that, of course, the entire US has problems in terms of forced insect pests, but the Northeast has a particular problem. So the authors of this paper went through and tried to decide or determine what was causing that. Why are we such a hotspot? And it's kind of a mix between the fact that, at least in terms of forests, we have a whole lot of different species of trees in the Northeast, more than most of the other parts of the country, although there are parts that have very unique trees relative to us, but we have a lot of different species. So that means there's a lot of different hosts here. That makes it a little bit easier to have a lot of different pests of those hosts. But the other thing is that we live, of course, in a part of the country that's highly, highly connected to the rest of the world through ports in Boston, New York, Philadelphia, and so on. And so we get a lot of global trade traffic through those ports. And along with all that traffic comes a suite of unwelcome guests, I suppose, in terms of invasive pests. And they have a lot of holes that they can take advantage of here. So we live in the hot spot. If it feels bad here, it's because it is. The other thing is to always point out is that these are not trivial concerns. You can kind of get in a little bit of a conversation about what the ecological impacts of all these pests are, and they can be quite substantial. But one of the more compelling arguments is always the monetary costs of invasive pest insects, both for agriculture, forestry, and infrastructure period. That's what this graph shows here. This is from a paper by Corey Bradshaw. And this is just the number of, the numbers by them are the number of studies associated with estimating those costs of pest insects across these different categories. I would argue, especially related to where we live in New Jersey and going on up into the Northeast, is that urban number is probably a lot higher. It's just that not a lot of people have measured or evaluated those costs in terms of invasive pests in that context. So we have a lot of pests, they cost us a lot of money, and they also have a lot of ecological impacts. So what are we supposed to do? What is our toolkit in terms of dealing with this problem? There's, of course, a lot of issues in a lot of places that you can attack the problem. But one of the most pernicious problems is simply finding these pest insects, especially when they are at very low abundance in the landscape. So if you are involved in any way in sort of trying to find an insect in a forest or an agricultural field or wherever it is, and especially when you're at the invasion front, right, the next place over from another place that already has them, and you're looking for them and going, okay, are they really here or are they not here? Do I have to worry about them or am I okay for right now? Then that feeling of, boy, I really wish I had a very strong survey tool to be able to answer that question. And in terms of the strength of the sensitivity of that survey, the technical term, what we're looking for mostly is to reduce the survey false negative rate. So this is when a survey fails to find evidence of the presence of an insect pest at a location where that pest is actually there. So that means that you looked for it, you didn't find it, but it is there. And so that's, in a lot of ways, the worst case scenario, because if you have it and you can't find it, then you've lost an opportunity to manage it in some way, or even in some cases eradicate. So most of the rest of what I'm going to talk about is centered around this problem. How do we reduce the false negative rate from the surveys for pest insects, both across agricultural and forest ecosystems? So that brings us to environmental DNA. So the definition of environmental DNA is it's genetic material that's shed by living organisms into the environment and can be sampled through the collection of, in most cases for environmental stuff, bulk samples such as water and soil. So environmental DNA basically allows us to detect the presence of any target species we're interested in, sight unseen. So all it has to do is leave enough genetic material around, and we have to be able to collect and analyze that genetic material to be able to say, yeah, it was there. So that's the main advantage, and one of the principal ways that we sort of can reduce that survey false negative rate is that we don't have to physically capture the target insect or target species itself to be able to say, yes, it's here. So this is actually very well developed outside of most agricultural and forestry applications. The two places you see it most often are actually in forensic science, where it's called touch DNA. And that's just the fact that every time you touch a table or wherever, you're leaving a little bit of your DNA there, which is a little bit frightening. But if you leave a little bit of that DNA there, and the forensic scientist can come through with, in this case, because it's very small and localized, they'll come through with a swab or something like that. And the DNA will move from that surface onto the swab, and then they'll take the swab back to the lab and hunt for a DNA signature. In this case, they hope to be able to find you in particular, because you have your own DNA signature and you're leaving it everywhere you touch. In terms of uses outside of forensic science, it's most used within any kind of aquatic ecosystem. So rivers, lakes, marine systems, ponds, whatever it is. And the idea here is that anything that lives in the ocean or any water, they are also shedding their DNA. So they're shedding their DNA the same way that you do when you touch something, but they also shed it in terms of their excrement. They shed it in terms of scales that pop off. They shed it in terms of eggs and other things that they do. So any living thing really is constantly shedding DNA into the environment. So if you go in and sample that water, the idea is that you can then look for the genetic signature of anything that was in that water and say, okay, it was there. So it's used now very regularly in terms of conservation. So we're looking for endangered species or tracking migratory species and when they come back, eradications of invasive aquatic things like mussels and whatever it is. And so just to make sure you really, really killed them all so you can get rid of them. It's used in fisheries in terms of just being able to get a better handle on what species of fish are in a section of the ocean that you would normally have to pull a trawl through, and it's sort of very expensive to do that, where you can instead just pull out some water and see what DNA is in there and figure out what is living in that water. The nice thing about water, and one of the things that's going to become important in a minute, is to understand that that DNA kind of mixes with the water. So once it's in the water column, it's going to move with the water column. And since that water is either in a river and it's moving around, or it swirls, or you have different layers of diffusion or anything that can happen within water, it will move and mix that DNA. So that's sort of a nice part of using eDNA in water, is that pretty much any pool of water you get from around the right area will pick up most of the species that are living nearby. So it makes it really quite easy. What we tried to do was take that technology for both the forensic part and for the aquatic eDNA and apply it in the context of invasive insects. So that takes it immediately out of water, so we don't have a water body that we're going to be able to sample. And that's not a huge deal, because there's a lot of work now on trying to, you can certainly get DNA out of soil. That's been known for quite a long time. So you can sample the soil and see what kind of DNA is in there. It's another type of eDNA. But other people have looked to survey for terrestrial species by just taking advantage of their behavior, like with these feral pigs, and they have these watering holes. So as long as you can get to enough watering holes, even if those pigs are not nearly, of course, as common as they are here, you don't need DNA to know that you have pigs right there. But you can go from water body to water body and be able to pick up their DNA, so you can detect terrestrial species in nearby water bodies. But both of these still don't really get us as far as we need to go in terms of making this a useful tool for agricultural and forest systems. So our big insight, I guess, and I'm sure that this came mostly because we began working with marmorated stink bugs and then later in with spotted lanternfly. And in both of those cases, the way the insect feeds produces a large amount of excrement. So we know that from spotted lanternfly. That's one of the reasons why they're so detrimental. They produce a lot of honeydew. But you have to then think about the fact that every time they're moving around and doing what they're going to do in terms of feeding, then they're leaving actually a lot of DNA on whatever it was that they were feeding on. So for brown marmorated stink bugs, it's any vegetables, it's peaches, it's anything that they're going after. And for spotted lanternfly, it's pretty much anywhere that they'll feed. They also will leave DNA the same way we do, through touch, and they will also leave DNA where they are related to shedding scales and other things. So, but we know, for at least for these species, that most of the DNA we're going to be able to pick up is because of the production of honeydew or a fair amount of excrement as they feed. So the question then is, how do you get that DNA off of those surfaces and then into a way that you can analyze it? So this is where we really kind of were pushing the boundaries and kind of able to do something really new and different. So this slide is just an overview of the challenge of eDNA sampling for forest or agricultural pests. So you have to start kind of thinking about, well, we have a lot of locations. Some of those locations will have our target pest species and some won't. And so we have to figure out which location it is. That's one of the main parts of the survey, of a survey at all. But let's say you're in location C and there is that pest insect there and it does leave eDNA there. How do you get it off of there? How do you get it off of that tree or those leaves or those fruit efficiently? And then where on that tree or that plant are you going to look? And then how do you move that into some sort of way that you can analyze it and go from there? So this was our solution. Our solution was to find ways to pull or aggregate the DNA. That's the main thing. How do you aggregate DNA that's left by these pest insects within a forest or within an agricultural field? And our solution, I'll show you some of the solutions that we figured out. There's several of them, and we're developing more of them even now. But the main thing is to basically pull them off of those surfaces where they have been deposited by your pest insect, aggregate them into water. So we'll put them into a water bucket that takes us into a water type environment. As soon as we get into water, then all that experience of dealing with aquatic DNA, eDNA, comes into play. So we don't have to reinvent the wheel from there. You take that water with all that aggregated eDNA, you end up concentrating it into a little filter, which I'll show you in a minute. You take that filter, you leave the water behind. You take that filter, which now has all the DNA that you think was out there, and take it back to the lab. And then at the lab, you extract the DNA from that filter, and then you run it through a qPCR, which is a standard sort of, these days, a rather old-fashioned way of quantifying how much DNA you might have in a sample. So that's the basic overall idea, and I would say that anything past the filter is pretty standard at this point. So I'm not going to spend a lot of time on that, but I can take some questions about the details of that later if you'd like. But the big step is, how do you aggregate the DNA? Oh, well, first I want to tell you another part of this, and that's when you get the DNA, how do you know you have the species that you think you do? So, okay, you can get DNA, and I suppose you can amplify it and extract it. But how do you know that DNA is from the species that you're interested in monitoring, controlling, managing, whatever it is? So the trick here is to pick a part of the target species genome that's exclusive to it and found in no others. So you find a gene sequence that is really only found within the genome of the target species that you're looking at. So this is as much of the DNA things and the details that I'll go into for the talk, but just to remind you, of course, that the DNA is just made up of a whole bunch of A, D, T's, and G's along the different segments of DNA. So what people in eDNA land have done is pick different parts of the DNA that they know are fairly exclusive to different species. So the most common one is to use a mitochondrial DNA, CO1 or CO2, and those parts of the genome are used in species barcoding, if you're familiar with that. So they do a fairly good job of distinguishing one species from another if you just sequence and get all the ACTs and Gs associated with CO1. We have been using also a nuclear piece of the genome, which is ITS 1 or 2. And again, we're just looking for sequences of ATCs and Gs that are exclusive to that particular species. There's kind of an accepted best practices associated with finding these pieces of the genome that work for eDNA. So we work through those. Those are, again, pretty well developed at this point because eDNA is so widely used, both in forensics and in aquatic ecosystems. So that's how you know, and you test. There's a testing you do to make sure that that's really a signature piece of DNA. So what we do is when we get that DNA and we pull it out, we're looking for that particular section, and that section tells us that we have just the species that we're looking for. So how do we aggregate the DNA from the field? So we have 3 different ways that we've used so far. So this is the first one that we developed. We did this, I don't know, because of how widespread UMass is, you might know the farm that we worked with, which is Heron Pond Farm, which is just right over the border from Amesbury, Massachusetts. They were very helpful in kind of giving us free run of their farm, their small vegetable farm. And we had the idea, we were hunting for brown marmorated stink bugs in this case. They weren't clearly in their fields, but we knew that they were close by. So we knew that they were probably, if they were there in very low densities. So we just waited for them to harvest the different produce that likely any brown marmorated stink bug is going to feed on. And then they bring it back to a wash station that they use. And so we just follow them and let them do the wash. And then any DNA that happened to be the brown marmorated stink bug DNA that happened to be on that produce would get put into solution into that wash station. And then we would just sample that wash station water. And then again, run it through a filter, take it back to the lab and see if we saw stink bug DNA. Well, that worked like surprisingly well. So we haven't done that much since, but it's there and out there so if people want to adopt that. You can do it for brown marmorated stink bugs. You can do it for any other species that you want where this might work in terms of deposition of DNA. It does rely on the farmers having a wash station, a wash step into their production process. So that limits a little bit. So for example, that's why we had to look for something different for vineyards. And certainly you can't do that for trees. But this works really well. So that's one way of doing eDNA for pest insects. The other one, which we've done in vineyards, is what we call spray aggregation. And that's effectively simulating rain and catching the rain. So you have, we have DI water or just fairly pure water that we can put into a backpack sprayer. So that sprayer is not pesticide, it's just water. And then little buckets that we have, and then you go through, and as long as you can stand close to whatever leaves or produce that you think that the pest insect is going to be using, in this case spotted lanternfly, then you just spray through there and then collect all the water that drips off all the grapes or the leaves, the grape leaves, whatever, or the vines themselves. So you just are kind of washing them and then catching the wash water. And this is nice because it's active. You can go out and do it. You can do it any time of the year. You don't have to wait for harvest. You can just go and do it whenever you want to do it. And it's pretty low tech, right? I mean, at least in the field, you're just kind of walking around spraying things and catching it in a bucket. Then once it's in the bucket in solution, you do the same thing in terms of running it through a filter and taking it back and seeing if you have evidence of in this case, spotted lanternfly. That still doesn't work very well for trees because a lot of parts of the tree you can't just wash off and catch the water. So in this case, we use something we call roller aggregation, which is just using a paint roller. You have to take the paint roller and make sure that it's completely free. You have to sterilize it basically before you take it out in the field, so just in case you don't want it to have anything else, any other DNA on it. And then you put it on a pole and you roll it across the tree. So this is very much a forensics crossover. So just think about those paint rollers as being like forensic swabs, and that you're just rolling it over a part of the tree that you can get to. Here we're hunting for spotted lanternfly again, so we're rolling the branches, we're rolling the stems. We've been working with the state of New York to come in behind them for a couple places where they think they might have it, but they can't find it, so we'll come in and do eDNA behind them. And so this is a New York City, actually I don't know what part of the city he works for, but he just went up in his bucket truck and rolled parts of trees in New York. We get a lot of funny looks for that, but looks like we're painting trees. But what we're doing is moving that DNA from the bark onto the roller, and then we take the roller back. Right now we're exploring how long that DNA will last on the roller, so maybe you can just stick it in a box and send it to the lab. But right now what we're doing is rinsing it off and getting anything that was on that roller into solution, and then moving it through our usual filters, etc. So for spotted lanternfly, we know that roller aggregation and spray aggregation work. So you can definitely pick up the genetic material of any spotted lanternfly that happened to be in that site and has left their DNA there. So they work fairly well. So that's a published paper if anybody wants to go look that up. So this is just a little bit of the steps so you can see. So that's a bucket that has water that was sprayed, collected, or using anything that I just talked about. You have a pump there that's on site. That's probably the most expensive piece of the field apparatus that you need. It's a peristaltic pump. And then you have a little filter holder on the end with the filter in the middle. It's a sort of etched filter. You have to buy those. They're not hugely expensive, but they're not dirt cheap. And then you move the water. You basically suck the water through that filter, and the filter catches all the DNA that happens to be in there. There's a lot of optimization that has to happen to get all this right, but including what size filter you need, but that's the general idea. Once that water's moved through, you don't really need it anymore, so you just dispose of it. And then you take the filter, put it in something that's going to be safe and secure and not pick up other DNA, take it back to the lab, and run the qPCR from there. And the qPCR on the other end, you can get two results out of it. One, you can say, yes, you have that DNA, or no, you don't. So just presence, absence of the DNA. Or you can get the concentration of the DNA on that filter, which broadly kind of gives you a relative idea of what the abundance of whatever target it is that you're looking for. So this particular slide, the one on the right with sources and sinks, is kind of complicated, but the main thing I want to say is that you have DNA coming into an agricultural system from spotted lanternfly or brown marmorated stink bug. And then it's also leaving that system, right? So there's a time frame in which you're going to have DNA that's there and is in a state that you can capture it and amplify it and make it run in a qPCR. In aquatic systems, this sort of whole process of how fast it's coming in and how fast it's going out, so how long it's there to be able to be detected, is well worked out. But in terrestrial systems, it's not. So we've had to do a couple experiments to see how long that DNA lasts. So for example, when a spotted lanternfly feeds in a vineyard and releases honeydew on vines or leaves, then leaves, and we go in a little bit later and are trying to find that honeydew, how long does it really last? Does it last years? Well, that's not great, because you don't want to pick up spotted lanternfly from last year. But if it's only there for a little while, then that might not be great either. So what we found is, not surprisingly, given that our methods work, is that when it rains, it pretty much washes all DNA that was deposited there off and into the soil in a way. So rain is a quick sink. So once that happens, you sort of wiped clean any DNA signatures that may have been left there. The other is just time and exposure to dry conditions and exposure to sunlight. And even if it doesn't rain, the DNA, amount of DNA that's left there by any single stink bug, for example, or a spotted lanternfly, will degrade through time. So after about a week, certainly by a week of nothing, once that DNA is there, after a week, it will degrade to the point where we can't really detect it. So if you pick up a DNA signature in one of our surveys, then you can be really sure that that means that there have been, in this case, spotted lanternfly or stink bugs there on that surface within, say, the past week. Okay, so I want to talk now about how, what are the applications of this type of survey in the context of pest insects. And I'm going to start with sort of asking, when would you not use eDNA for pest insect surveys? When the density of those target pest species is high enough that their presence is obvious, you would never use eDNA, right? So this is not going to replace a lot of the IPM programs, et cetera, that that are employed for pest species that's sort of already there, and you're responding to it. So, for example, we haven't used brown marmorated stink bug, our application there very much, because at least in New Jersey, it's so widespread that you don't really need eDNA to tell you that it's there. So the same is going to be true for spotted lanternfly, that when you can walk out and see them, because they're not very difficult to see, funnily, I mean, they're not small. And so if you can see them, you're not going to use eDNA. Same with emerald ash borer. If you have a tree that has this many exit holes, you don't need eDNA to tell you that you have emerald ash borer. So we're not talking about using this survey in the context of something that's pretty widespread and abundant. But you may want to use eDNA surveys in any situation that's characterized by the pest population being in low densities, or when they're very cryptic and hard to find, or when accessing a location can be dangerous or really expensive. So this particular little graphic I did was for coconut rhinoceros beetles that are a pest in the Pacific on palm trees, but it applies here as well. So you can we can see applications for this within biosecurity, and that's usually either low density or cryptic, and that you're looking for them within different products, you're looking for them around ports, you're looking for that first entry into a continent, into a country. So you can definitely use them in that context. You can use them to estimate relative abundance, especially in places where accessing that site can be relatively difficult. But the ways we're thinking about it, and what I'm going to show you mostly for the next several slides, is how you would use it to find little satellite populations away from the place where you know they are. So you're trying to control the spread. Surveys, where you really need to find the very front edge of that pest species range, so that you can kind of fight backwards and start at the very edge of where they are and move back in and try to control that spot. So those are related. So if you find a satellite population, you want to delimit it, and then you want to attack it in terms of whatever management options you have. But those are two situations where sort of conventional sampling are challenged by the fact that they are tasked with finding something, a pest insect in the landscape, when it's very, very rare. So it's a hard lift for any survey. And then I also like to talk about it in terms of successive eradication, stop the spread program. So this is really the most quantitative slide I have. And the top graph on the left is just the probability of eradication. So 1 is a successful eradication, and 0 at the bottom is an unsuccessful eradication. And the bigger the circle, the more examples there are. And then along the x-axis, you can see that's log area. So that's the infestation level, going from a really small infestation level on the left to really large on the right. And then the bottom one is just sort of the same x-axis, but we have the cost of that eradication attempt plotted against that area of the initial infestation. So the big thing here is to note that you have a lot of big circles when the area of infestation is small. So eradication is far, far more likely to be successful when you catch an infestation when it's very localized and small population size. It's not that you don't have success when it gets large. You do, but they're much less common. And then the bottom one shows you that, yeah, you can achieve some success, but it's going to cost you a pretty penny. So that cost is in millions of dollars in a log scale. So really, if you're talking about how to super efficiently respond to the really large number of pest insects that we have in the Northeast, you really want to be on the left side of these graphs. You want to be, when the infestation is small, it's easy to control or even eradicate, and it doesn't cost you a ton of money. The other graph on the left here just indicates the enormous advantage you get from having a really good survey system. So this is the number of programs versus successes and failures, for the black as successes and gray as failures. And detectability is just a kind of reverse of survey false negatives. So a high detectability means that you don't get a lot of false negatives. So when you have a survey tool that has high detectability, meaning that you can find something even when it's really rare, you have a lot more successful eradication attempts. And then on the other side, it's that method of detection, so active, passive, and search. And the big difference here is that active means that you go out and you decide where you're going to survey, and you can survey that area, and you can do it lightly, you can do it exhaustively, you can control that. Where passive is just sort of putting something out and waiting for the insect to kind of come to you or come to it. So if you have an active method with a high detectability, you're much, much more likely to find those populations when those infestations when they're low, small, and then your success is high, and your cost of doing that eradication or that control is low. So that's kind of what we're aiming for: high detectability, active methods, get them when they're rare. So that takes me around to what we've done with spotted lanternfly. If you're not familiar with spotted lanternfly, you probably will be soon. So this is a pest insect that's from Asia. It showed up, oh boy, 4 years ago, initially in Pennsylvania, and then has spread out from there. So it's they can get to really large numbers, like, biblical plague level numbers when you're walking through. It gets a little weird. But the main problem that they have is, one of the main things is they are generalist in terms of what hosts they can feed on. So they are in Tree of Heaven pretty much year round, which is another non-native species here. They do group around Tree of Heaven when they're ready to breed. It's not clear why that's happening, but they also will, you could definitely find them on walnut. And then in terms of agricultural costs, they use wild and cultivated grapes pretty much through their entire lifespan. And they can do some serious damage to vineyards for sure. It's not as clear exactly what kind of damage they're going to do in terms of forestry. But it's not uncommon for a vineyard that gets a high infestation level of spotted lanternflies to lose their plants will die. So they'll have to. It takes a couple years for that to happen, but they will lose part of their crop, which in terms of grape or anything else, it takes years to actually get established as a huge loss. So there's a lot riding on finding where these guys are. So this is the latest map of where spotted lanternfly are for sure in the quarantine areas. So they are definitely in New Jersey, but you can see where they started in Pennsylvania. And then they've moved in, not surprisingly, in a little bit to Delaware and Maryland, and they are also in the West Virginia area for sure. So they are moving out and spreading across. And I'll say that this is probably a year, we're going to see for sure, Rutgers is sort of at the little bendy part of New Jersey, and we already see them on campus. So they're getting into that New York Harbor area, which is not good for the rest of the world, I have to say, because we're going to end up accidentally exporting them out of here. But they are on the move, so I don't know what will ultimately limit how far they will go in terms of natural limits, but they are definitely moving around. So the existing survey tools for spotted lanternfly are just visual surveys, but also the sticky band traps, which you can see at the bottom here, and also this is a newer one of these kind of passive funnel traps. So they do, especially the nymphs will crawl upwards, and so if you can kind of catch them and funnel them into a particular area, you can capture them. These are both, the sticky band traps work pretty well, but they work too well. They also have a lot of, bycatch, if you will. Anything that touches them will get stuck to them, so there's people don't really like to use them because they'll catch birds and squirrels and things, which is not what we want to happen. So people are moving to these funnel traps, but they're still kind of passive traps. There's no known lure that works. They're trying to develop it, but so far there's nothing that works particularly well in terms of luring spotted lanternfly to some of these traps. So we're stuck with kind of a passive trap that doesn't even have a lure. So we don't have the bass survey tools at the moment. So this is where we came in because we developed this spotted lanternfly eDNA technique. We've tested it just proof of concept across urban forests and a few other places. But last fall, we went into vineyards in New Jersey and conducted a couple trial runs of what this looks like and how it compares to just doing a visual survey. So this is us in the field. This is one of the best field experiences I've ever had. And walking around a New Jersey vineyard is great, you know, especially the right time of year after you're done with the surveys. You just drop by and have a tasting. You know, that's a nice way to do field work. But the goal here was for us to go through and do several plots, do our spray aggregation, and do visual surveys, so trying to find them at the same time. Last fall, New Jersey was a great time to do this because we knew they were there or they were close to there, but they really weren't in high density. And in fact, we took advantage of their behavior. So the times that we went into a time when the lanternfly hadn't moved out into the vineyard yet, so there really were hardly anything in the vineyards. And then we went out a little bit later in the year when they came off of the breeding aggregations and they moved back into the vineyard. So we have two sort of natural lanternfly densities to play around with. So here's just a little bit of our results, and that this just shows the number of lanternfly we had in visual surveys and the amount of DNA we had in our DNA samples. Round 1 is low density time of year, round 2 is high density. So there's sort of two take-home messages here. One is that as spotted lanternfly visual survey detections go up, the amount of DNA that we pull out goes up. So they broadly sort of match each other. The other one is if you look down on the 0 part for visual surveys, that's when we found 0 visual detections of lanternflies, but we had a fair amount of detections with the eDNA. So you can see that we are picking them up when visual surveys would not. If you translate this into detection probability, or the chances that you're getting a false negative, then you can see that visual surveys have a really high false negative rate, which here is kind of a low detection probability. So if they're there, or even given that they're there, the chances that we find them are pretty small, like under 10%. For eDNA, given that they're there, the chances that we find them with eDNA go up to around 60%. So that's a massive improvement at low densities. And even at high densities, when we were picking up many more visual surveys, we were picking up much more spots and plots that had spotted lanternfly. So we were up near 100% detection probability, whereas visual detection was still down around 50%. So this is the first sort of clear data that we can show that eDNA in this case is really reducing our false negatives relative to at least visual surveys. We haven't compared it yet to those traps. We had it all set up to do that this year in West Virginia, and to work with the USDA folks there, especially in terms of figuring out like the limiting advantages that we can have. And then of course everything was shut down. So we still hope that we'll get out to do some of that, but I think some of it's going to have to wait till next year. Okay, so I'm going to sort of start to wrap things up with talking about how we're trying to expand this a bit. So we've been working with these insects that release a lot of honeydew or just a lot of excrement. How does it work when we get into other known pest species? And so that don't do that. So we've started working on emerald ash borer, and it presents a whole new challenge, which is fun from our point of view. And that what we think we'll be able to do is that when the adults are out and feeding on the leaves of ash trees, they release a lot of frass. You can see that in kind of the lower right. Can we pick up that frass from some of our techniques, or do we need to develop new techniques? But that's going to be DNA that's going to be there and left there after the adults are gone. So if we can find a way to aggregate that kind of DNA, then we can tell you whether or not there's adult emerald ash borer feeding in the canopy of a tree, and hopefully we can pick it up at low density, so you can know that you have an infestation in a tree long before you would ever see all those holes. And then really a challenge is to see whether or not we can detect the boring phases of emerald ash borers, and in general, can we pick up anything relative to wood borers using eDNA? And that's a real challenge as well. They don't leave a lot of DNA. Is there, we have to, that's one that we're trying to tackle right now, is like, how would we pick that kind of DNA up? So we have, we're right on the cusp of finishing off an assay that tells us that this is in fact a chunk of emerald ash borer DNA that's specific to them. Doesn't indicate any other species presence, so we're close to getting that. So then that lets us move into trialing out all these different possibilities relative to detecting emerald ash borer DNA when they're very rare. This is a couple years old, but you might, in New Jersey, we sort of like, oh, well, it's here now and it's not going away. So again, I'm not sure that you would use eDNA in that context, but there are several places where emerald ash borer are expanding into, where you would want to be able to use it, including up into Maine, possibly places in New York, but any place you see this sort of one-off little county sign, that like Colorado, that you would want to be able to go in, see if you have it, find all the places that you have it, especially if they're at low density, so you can gauge your management reaction accordingly. Another sort of piece of this for emerald ash borer is to be able, one of the nice things about eDNA is that you can decide when you're doing this, you're pushing all this DNA into water, into a bucket. So you can decide how much water you want to put in there from wherever you want to do it. So if you're sampling a whole forest, for example, then you may do the roller across all those trees and then just rinse them all into one bucket. And if that's the case, then you're making a sort of an inference about that forest. But you could also roll just one tree and put that roller into one bucket and have that. So you can get information from a whole forest, or you can just choose to do it for one tree. So if we can figure this out for emerald ash borer, then it would allow us not only to say, okay, this is a forest that probably has emerald ash borer, even though in the canopy and they're rare, so you can't quite see them yet for normal surveys. But we can also go through any kind of localized place where you need to know this tree has emerald ash borer, these sets of trees, or this tree does not. So you can get it down to the tree level. And hopefully, because there are treatments for trees associated with protecting them against the damage of ash borer. So if you can identify a tree that's in the early stages of an infestation, then you can catch it and respond to it and do the injections and the everything that you need to do to protect that tree early. So we see a lot of possible applications in terms of that, in terms of forests, urban forests, any forest really. Okay, so I'm going to end up on what are the costs. So the cost in terms of field collection is low. I mean, it's really low tech. It doesn't take much to go to Home Depot and get a couple sprayers in a bucket. There are some, you have to have a level of OCD to make sure that you don't contaminate your samples, because the DNA moves so easily from one surface to another. But beyond these things and having the right protocol, really the most expensive thing you might need is that pump, which is about $1,000. So that's the cost in terms of the field. The number of people you have out really is up to whatever your goals are, and you can have one person doing this, that's fine, or you can have an army of folks trying to do this over a larger area. The lab part is the part, of course, that adds the costs, and they're like we do. My lab does some. We will support some applications like we do in New York State, and it's about $20 per sample for us to do the lab part. That includes a whole bunch of university overhead and all the rest of it. So if people develop the ability to do the lab part in-house, and again, there's nothing really revolutionary about the methods that we're using. qPCR is pretty well trod at this point. Then you can lower that cost even more. There are also companies that have sprung up because of eDNA uses across fisheries and aquatic systems and everything else, that they will do it for you. So you can go to a private company and just contract with them to do the lab part. is the sort of cost worth it, and how does it compare? Well, that's one of the things that we're hoping to directly address and calculate all those costs in terms of spotted lanternfly and vineyards over the coming year. This is some of the preliminary data we have showing, what is the cost if you calculate it as how many sampling plots do you need to get to some sort of detection probability, and so in this case, that gray line is a 95% confidence that you really do have lanternfly in that vineyard. And then the bottom is the number of plots you had to sample to get to that 95% confidence. And the one on the left is the low density version, and the one on the right is the higher density version. And you can see that in both cases that eDNA curve is shifted to the left, which means it's going to take you to the 95% confidence interval with fewer sampling events. And even when, for the low density one, you have to see that the main thing to see is that we can get to a 95% confidence that they're really there with about 10 samples, where the visual surveys, where you kind of get there right at the very end of about 30 surveys. Again, we're going to go in and see how much that changes with the traps, the funnel traps. But even in a sort of higher density situation, we can tell you that eDNA can tell you that you have spotted lanternfly after like 1 or 2 samples, whereas even with visual surveys and them being a little bit easier to find, you'd be up near 15. So the cost is going to be related to how much savings you get by how much ground basically you have to cover with sampling effort to get to some sort of goal in terms of your survey. And it's going to, the payoff is going to be how valuable is this information that you have the insect, the pest insect there, so that you can manage and get to that eradication and the control in a more cost-effective and more successful way. So there's a lot of cost elements to this, but this is some of the basic pieces of that. So I have a lot of people to thank. We're funded through the New Jersey Department of Agriculture, the SARE program, McIntyre Sinus at NEFA, and then I have a whole army of people who've kind of helped out through the years, including Ann Nielsen, who's our fruit entomologist, and several people from my lab. So I think with that, I'll throw it back over to Tony to help with questions.
Excellent. Well, thank you so much, Dr. Lockwood. That is fascinating, and I have so many of my own questions, but we have a lot of questions from the audience, so I want to focus on theirs, but very cool stuff. So the first question I have is from Andy. What is the impact of strong water currents on sample dispersion and detection of main habitat? And he asks about following the currents. So I assume this is in aquatic systems?
Yeah, so there's a lot of research on that. It moves pretty quickly. So when you have a strong current, so a river, a fast-moving river, any DNA that's deposited by a fish, unless there's an eddy or something there that's going to hold that water for a period of time, it's going to flush on down. So that's a good point. And that's one of the issues with aquatic DNA is in a river like that is if you pick up DNA, are you sure it's really from that location, or do you think it's upstream? So that's been a persistent issue in trying to figure that out for a while.
Excellent, thank you. A question from Kathleen. So they have been able to take a spotted lanternfly and map its DNA, and then they can in turn look for their DNA in the water or fields. So maybe more about a question about your process.
Right, so the DNA is, obviously the spotted lanternfly's DNA is in their honeydew, and then when they on a leaf or a vine or a grape, then it stays there, and then they can move off. And then what we do is come through and basically wash that honeydew off just with water and collect it in a bucket. And then now that DNA is in solution in that bucket. So that's where we're moving the DNA off the surfaces and then into water. And then once it's in the water, we move it to the filter. And once it's in the filter, we can take it back to the lab efficiently. So same thing with trees. So a tree where a spotted lanternfly happened to be feeding, and they will be on any branches. You can see them pretty regularly. then if they leave, or they drop from the upper canopy, if it just drops down and happens to land in understory or on the tree itself, then we can just remove that DNA, which is basically the honeydew, we think, and using those techniques, put that DNA into solution that allows us to filter it out and take it back to the lab.
Excellent, thank you. A question from William is, excuse me, is the eDNA test qualitative, basically presence or absence, or is it also quantitative? So can you tell the general size of a population?
It's both. So what you get out of the qPCR is an amount of DNA, and what we usually that sample had, and so we usually put a little lower limit on that to say if it had at least this amount of DNA, we'll say that it's present, and if it had less than that or there was no DNA in there, it's clearly absent. But you can also just track the amount of DNA in there, and so the question is, for that part, is it's hard to take that amount of DNA and then pin it back to, oh, you have 10 or you have 100 spotted lanternfly, because there's behavioral issues with the, how much honeydew are they producing? It's the honeydew production, which is obviously going to go up with more spotted lanternflies, but it might go up behaviorally or other things as well. So you can kind of get a rough relative idea of abundance across different places, but I would hesitate to ever pin it on, if you get this amount of DNA, you have this many spotted lanternfly.
Thank you. Another question from Andy. This is about lures, and I think possibly for spotted lanternfly, conquered grape juice as a lure, given their association with grapes during their whole lifespan. Is anyone looking into that?
Well, that's a good question. I'm not sure. The USDA lab out in Cape Cod is doing a lot of different possible lures. And last I heard, they had something that seemed to work okay, but like which I think would be equivalent to just having the juice chemical odor around. But it wasn't like there doesn't seem to be like a sex pheromone or something that attracts them all to one place. So yes, I can't say for sure, but they have been playing around with lots of different possible chemical cues in the environment to see if they will respond.
Excellent, thank you. A question from Kathleen. She says, so going back to the eDNA test results, should the eDNA be used in conjunction with the visual inspections to confirm, or should it be used exclusively because sometimes the visual inspections show false positive results or false negatives?
Well, actually, so eDNA will show, but you have the possibility of both false negatives and positives, so you have to be aware of that. That's true with eDNA in general. There's some things that you can do to reduce both of those, but especially with handling with false positives. That's where I said you have to be kind of OCD in the field and in the lab so that you don't generate those. But so far, I should say that it's very rare that we've seen anything in our work that produces those. So that's an interesting question, because, and it's not resolved, because here's the deal. Like if I say, hey, you have spotted lanternfly based on DNA evidence, does that give you right, does that fit the regulations that are required to use a pesticide? Well, probably not. Does the state really want to, any state agency really want to jump on just an eDNA detection? So there's lots of rules and regulations associated with establishing quarantines and all kinds of things like that that don't recognize a DNA hit. Will they in the future? Possibly. Do they need to be updated for this? Possibly, but I'd say that's probably at least 5 years out. So when we work with New York, for example, we'll give them our results, and then they go in right after us and then just blitz it in terms of a visual survey, because they can't do anything until they have a visual confirmation. So we give them a heads up and help them kind of narrow down all the different places in New York where they potentially have an early infestation, and then kind of give them a little, I think about it as like a red, a yellow, or a red, green light. Meaning green light, we didn't get anything. you didn't get anything, it's probably okay. A red light being, wow, we got a lot of DNA. You really need to go in and see if you can find it visually so you can move on. And then a yellow is that kind of in between where we got some DNA, it wasn't a ton, it passed our detection, you know, our criteria, but it wasn't like we got a whole lot of hits. So, and then they just decide it's up to them really to decide how they want to respond to those signals that we give them.
Excellent. Well, thank you. This is fascinating, and I apologize to the folks that we have run out of time to ask a couple more questions. So I will switch over. I can't thank you enough, Dr. Lockwood, for your time and for your discussion today. We need to switch over to the instructions for folks looking for association and pesticide credits at this time, so I can go over that once we can get the slide up. All right, perfect. So for those of you looking for association credits, again, a reminder to the Mass Certified